So, hello everyone. Today we will talk about the endoplasmic reticulum. So, this is a membranous structure that surrounds the nucleus, and this is generally involved in synthesis of proteins and lipids. If we look at the ultrastructure of the endoplasmic reticulum, so this is a membrane that continues with the nuclear plasma membrane. So you can see the outer membrane of this nuclear envelope. This continues into these pouch-like structures, what we call cisternae. So the endoplasmic reticulum consists of these pouch-like structures or tubules. So cisternae means it means a reservoir for liquid. So because the soluble proteins, they reside inside these pouch-like structures, and these pouch-like structures, in case of rough endoplasmic reticulum, which is a type of endoplasmic reticulum, this contains these dotted structures. These are basically ribosomes. So there are two types of endoplasmic reticulum: smooth endoplasmic reticulum and rough endoplasmic reticulum. Why do we call it an endoplasmic reticulum? So endoplasmic means within the cytoplasm, and reticulum is the Latin for little net. And so it is basically a little network that is found inside the cytoplasm, and it surrounds the nucleus, and this contains these pouch-like or tubular structures which are filled with liquid. So that's why they are known as, these pouch-like structures are known as cisternae. Cisterna is the singular and cisternae is the plural. So we have these two types: smooth endoplasmic reticulum and rough endoplasmic reticulum. So we will first talk about the smooth endoplasmic reticulum and then the rough endoplasmic reticulum. If we talk about the functions of the reticulum, so there are two major functions. One is a synthesis of lipids or steroids, and the second one is the synthesis of proteins. So the smooth endoplasmic reticulum is generally involved in the synthesis of lipids, while the rough endoplasmic reticulum is involved in the synthesis of proteins. And as we have seen, that the rough endoplasmic reticulum, that is studded with ribosomes. So these ribosomes are sitting on the outer side of the rough endoplasmic reticulum. So these are the enzymes that these are the ribosomes that are involved in protein synthesis. So there are two types of there are two regions where protein synthesis takes place. One is inside the cytoplasm. So here the proteins are synthesized by the free ribosomes inside the cytoplasm, while the other proteins, these are synthesized on the rough endoplasmic reticulum. Because of the presence of these ribosomes. So these are the ribosomes that are sitting on the outer side of the rough endoplasmic reticulum, and these are involved in the synthesis of proteins. So generally, there are exceptions, but generally the proteins that need to stay inside the cell, they are synthesized by the free ribosomes inside the cytoplasm, and those that need to be exported out of the cell, these are generally synthesized on the rough endoplasmic reticulum. You can also see that the outer membrane of the nucleus, this nuclear envelope, that also contains some ribosomes on it. If you talk about the structure of the endoplasmic reticulum, we have previously discussed that it consists of pouch-like structures that are cisternae and tubular structures. So this is basically a very large membranous structure which accounts for 50% of the total membranes in a eukaryotic cell. So this is the amount of membrane found inside the cell. If we put together the membranes of all the other organelles, like mitochondria, Golgi apparatus, lysosomes. So if you put all these things together, so the endoplasmic reticulum alone accounts for the same amount of membrane. So it's like 50% of the membrane inside a cell is only the endoplasmic reticulum. And so that's more than half, or at least half of the total membranes. And if you talk in terms of space, how much space is there, uh, as we have discussed these are pouch-like structures, and these are reservoirs of liquid. So how much of the stuff can these pouches store? So it's one continuous space. So this is a continuous space. So there are no breaks in between, like there are. So the pouches, the Golgi apparatus also contains these pouch-like structures, but they're not connected to each other. So in this case, the endoplasmic reticulum, that is connected. So all the sacs and tubes, they are connected to each other, and this is a single continuous space which accounts for 10% of the total cell volume. So the smooth endoplasmic reticulum, that is basically involved in the synthesis of lipids, and the rough endoplasmic reticulum, that is involved in the synthesis of proteins. So both the soluble proteins as well as the transmembrane proteins. So the soluble proteins, or water soluble proteins, these are found inside these sac-like structures, so they're free-floating. And the transmembrane proteins, these are synthesized on the endoplasmic reticulum, but they stay embedded inside the membrane of these endoplasmic, rough endoplasmic reticulum. And when the vesicles are exported out of the cell, so the soluble proteins, these are released out of the cell, and the transmembrane proteins, which were embedded into this membrane of the rough endoplasmic reticulum, they stay inside the plasma membrane as transmembrane proteins, because they're bound to the membrane, so they cannot be released. So there are two types, smooth endoplasmic and rough endoplasmic reticulum. The differentiation, whether we call it smooth endoplasmic reticulum or rough endoplasmic reticulum, is not because of their function. It's rather because of their appearance under the microscope. So if you look at these two types of endoplasmic reticulum, the rough endoplasmic reticulum is called as rough endoplasmic reticulum because it appears rough, and it appears rough because of the presence of the ribosomes on top of it. While the smooth endoplasmic reticulum, this does not contain any ribosomes associated with its membranes. A rough endoplasmic reticulum contains ribosomes because it is involved in the synthesis of proteins, and smooth endoplasmic reticulum does not contain ribosomes, and this is why it appears smooth under the microscope. So let's talk about the functions of the smooth endoplasmic reticulum first, and then about the functions of the rough endoplasmic reticulum. So the smooth endoplasmic reticulum, it's basically a factory which processes lots of things, and it's involved in many metabolic processes like synthesis and hydrolysis, or breakdown of lots of different stuff. And this happens because of the presence of different enzymes that this smooth endoplasmic reticulum contains. So this is involved in the synthesis of lipids, oils, phospholipids, steroids, and sex hormones like testosterone, estrogen, progesterone. So that's a factory that synthesizes lots of these lipid-based things. And then it is involved in the hydrolysis or breakdown of glycogen in the liver into glucose. So glucose is stored inside the liver in the form of glycogen, and whenever there's deficiency of glucose, like you have not eaten something for a long time, or if you have been sleeping at night and you wake up early in the morning, so what liver does, it keeps converting glycogen into glucose and keeps pouring this glucose into your bloodstream, so that when you wake up early in the morning, you're not hypoglycemic. There's also a phenomenon associated with this process. It's known as the dawn phenomenon. This happens very early in the morning, that your blood glucose levels rise. So this specifically happens in diabetic patients, because their liver is producing more glucose from glycogen. So these patients may have slightly increased blood glucose levels early in the morning. And then it is involved in the detoxification of various drugs and poisons, because the smooth endoplasmic reticulum, and it's also involved in the metabolism of alcohol and barbiturates. It contains it contains different enzymes like cytochrome P450. So it's a very famous enzyme. It's cytochrome P450 is a superfamily of enzymes which oxidize fatty acids, steroids, and xenobiotics. And they also play a major role in the clearance of various compounds from the liver or from your body. And then they're also involved in the synthesis of various hormones, and they're also involved in their breakdown. So all those cells which produce the lipid-based enzymes, they are supposed to have larger amounts of smooth endoplasmic reticulum, because this is the structure that is involved in the synthesis of hormones. Or when you have when you're taking drugs, so some drugs can also influence the function as well as the structure of the endoplasmic reticulum. For example, if you have been drinking alcohol, or if you have been using barbiturates, so these are sedative pills, they make you sleepy. So what happens, we often observe that you start with the smaller doses and then you need larger doses later. This happens because of the phenomenon what we call drug tolerance, and this happens because certain drugs and alcohol, so they induce proliferation of the smooth endoplasmic reticulum. So both smooth endoplasmic reticulum as well as rough endoplasmic reticulum, they increase their content. So they can increase in size, they can increase in volume, depending upon the demand. So if you're so certain drugs, when they induce proliferation of the smooth endoplasmic reticulum, so now you have more smooth endoplasmic reticulum inside the cell. So the larger the smooth endoplasmic reticulum, the higher would be the number of enzymes which are involved in the breakdown or detoxification of the drugs. So what happens that these drugs, you start with one pill a day, and after some times this is cleared from the liver, or it is metabolized by the smooth endoplasmic reticulum faster because of the higher number of enzymes present inside the smooth endoplasmic reticulum. So you have been taking drugs, the smooth endoplasmic reticulum keeps proliferating, it keeps increasing in size. So now you have a higher number of enzymes present inside the smooth endoplasmic reticulum to metabolize or detoxify the same amount of drug. So what happens that a drug which had a half-life of 12 hours, it is left with a half-life of 8 hours. So what happens that gradually you need to consume more pills in order to fall asleep, as compared to what you started with. And the same is true for alcohol, that with the passage of time your satisfaction level needs higher amount of alcohol as compared to what you started with. I have added some text, you can read it yourself. I have added this text, you can read it yourself later. So this basically talks about the same things. Another important function of the smooth ER is calcium storage. So liver contains a specific modified form of smooth endoplasmic reticulum. This is known as sarcoplasmic reticulum, and this is involved in the storage of calcium. So what happens that I don't have the structure. So anyways, what happens that the smooth endoplasmic reticulum, it keeps collecting calcium from the cytoplasm into its tubules, and in most of the cells, the smooth endoplasmic reticulum has specific regions for calcium storage. So what happens that this membrane contains calcium pumps. These pumps pick up calcium from the cytoplasm and store it into these specific regions inside the smooth endoplasmic reticulum. And inside the smooth endoplasmic reticulum, we have calcium binding proteins. So calcium remains bound to these proteins, and this is released quickly in response to external signals. And if you talk about the sarcoplasmic reticulum in the muscle cells, so the quick release of calcium from the endoplasmic reticulum into the cytoplasms leads to contraction of the muscles, and bringing back the calcium into the endoplasmic reticulum, smooth endoplasmic reticulum, would lead to muscle relaxation. So this is also a very important function of the smooth endoplasmic reticulum. Okay, I think that's sufficient for smooth ER. Let's talk about the rough endoplasmic reticulum. As we have discussed, this is called rough because of its appearance under the microscope, as it is studded with lots of ribosomes. So most of the proteins which are needed to be exported out of the cells, what we call secretory proteins, so these are synthesized on the rough endoplasmic reticulum. And as we have discussed, that smooth endoplasmic reticulum would be abundant in those cells which are involved in the synthesis of lipid-based or fat-based hormones or steroids, like testosterone, like adrenal glands, or testes that produce adrenal glands, or ovaries. So it would be more in the ovarian cells, which are involved in the synthesis of estrogen and progesterone. While rough endoplasmic reticulum would be abundant in those cells which are involved in the synthesis of proteins like thyroxine. So thyroid gland produces thyroxine, like you have plasma cells which produce antibodies. We have pancreatic acinar cells which are involved in the production of digestive enzymes, or we have these islets of Langerhans inside our inside the pancreas, which produce insulin and glucagon. These are two famous hormones which are involved in the regulation of glucose metabolism. So all these cells which produce protein-based hormones, they would have large amounts of rough endoplasmic reticulum. So what happens is that the mRNA is released from the nucleus through the nuclear pores into the cytoplasm. So if this protein is needed somewhere in the cytoplasm, so that would be synthesized by the free ribosomes inside the cytoplasm and would generally stay inside the cytoplasm. Although there are exceptions where proteins move back again into the endoplasmic reticulum. We will discuss it later. But most of the proteins that are needed to be exported out of the cell, these are synthesized by these ribosomes which are sitting on top of this rough endoplasmic reticulum. And these ribosomes, after synthesis, they keep pushing these proteins into the lumen of cisternae. They're sitting on top of the endoplasmic reticulum, and they have a hole next to them, and they keep pushing these proteins through these holes into the cisternae, these pouch-like structures. And then these pouch-like structures, so at the end we have this transitional endoplasmic reticulum, because this can transform into, or this can produce these vesicles. So part of the membrane is detached or from the endoplasmic reticulum, from the rough endoplasmic reticulum. It develops into a vesicle-like structure which contains both soluble proteins inside this vesicle and transmembrane proteins which are embedded into the membrane here. And later this vesicle fuses with the Golgi apparatus where proteins are processed, and then after processing, so the post-translational modifications take place inside the Golgi apparatus. So it passes through these different pouch-like structures, and at the end again the Golgi at its trans end. So both the endoplasmic reticulum and the Golgi apparatus have these two ends. One is cis and the second one is trans end. So for the endoplasmic reticulum, cis end would be the end that is closer to the nucleus, and trans end would be the end that is the one that is at the periphery. And the same is true for Golgi apparatus. Cis Golgi is the Golgi which is lying closer to the endoplasmic reticulum, or this is the receiving end, and trans is the trans Golgi is the region where the vesicles bud out. So that's the periphery or the distant region of the Golgi apparatus. So these vesicles then later fuse with the plasma membrane. So they have all of these membranes have the ability to fuse with each other because they have the same composition. They are phospholipid bilayers. So these phospholipid bilayers, because of having the same composition, can fuse with each other very easily. We will study this later, how these transitions take place and how these vesicle-like structures fuse with each other. So ultimately this vesicle that contains these proteins which are needed to be exported out of the cell, be it insulin, be it glucagon, be it thyroxine. So all these proteins that needed to be exported out of the cell. So the vesicle fuses with the membrane, and it exports out these soluble proteins, while the proteins which are part of the plasma of the membrane of the Golgi apparatus. So because they are embedded into the membrane, they later stay embedded into the plasma membrane, and they work as transmembrane proteins inside the plasma membrane. So I think that's it for endoplasmic reticulum. Thank you so much.